Good morning, everyone, or good afternoon, based on where you are joining us. And welcome everyone to today's PGC ISPG Worldwide Lab webinar. I'm very pleased to chair today's session on integrative multi-omic approaches to understanding PTSD together with Jessica Dennis. This webinar is part of the new Worldwide Lab series jointly organized by the Psychiatric Genomic Consortium and the International Society of Psychiatric Genetics, with the goal of bringing together researchers from across the world to share new findings, methods, and perspectives in psychiatric genetics. Today's session highlights the growing importance of integrative approaches in PTSD research, particularly efforts to move beyond single-layer biological associations toward integrative models that connect molecular, genomic, and neural mechanisms. We are very fortunate to have three outstanding speakers who will each approach these efforts from a complementary perspective. Our first speaker is Dr. Matt Girgenti from Yale University. He's a neuroscientist and molecular biologist whose work uses human brain tissue to study molecular and genomic mechanism underlying psychiatric disorder, including PTSD, depression, and alcohol use disorder. He will present work on chromatin accessibility and 3D genome organization in PTSD. Our second speaker is Dr. Nikolaos Daskalakis from Boston University, where he is professor of pharmacology, physiology, biophysics, and medicine biomedical genetics. He directs the Neurogenomics and Translational Bioinformatics Lab and is a founding leader of the PGC-PTSD Systems Biology Group and PsychENCODE PTC BrainOmics project. His talk will focus on systems biology approaches to dissecting PTSD across omics layers, brain regions, and cell types. And our final speaker is Henry Bailey, a PhD candidate in biostatistics at Boston University. His research focuses on understanding the genetic architecture of psychiatric disorders. Today, he will present work on shared genetic influences between PTSD and resting state brain networks, with a focus on how PTSD-linked genetic variation may relate to spatial temporal synchronicity across major cortical networks. So for logistic-wise, each speaker will present their work and we will have time for discussions and questions at the end of the webinar. Please free to submit questions through the Q&A box. We will be monitoring the chat and direct the questions to the speakers. With that, I'm delighted to welcome our first speaker, Dr. Matt Girgenti. The stage is yours.
Thank you, Seema. Can everybody hear me? Okay, good.
Yes.
Because I see that there's a second version of me in the Zoom, so I just want to make sure. All right, let me check. And hopefully everyone can see my talk.
Yes, we
Okay,
can see.
great. Thank you. And again, thank you for the invitation. It's really exciting to be here and to speak with all of you about the work that we're doing at my lab at Yale. I'm going to be showing you kind of a combination of some previously published work that we've done in addition to some unpublished work that I think is really exciting and really ties together the themes that the planned webinar was for, including looking at multi-omics across patients with PTSD. So let me jump right in and talk a little bit about chromatin and how the chromatin environment is affected by individual SNPs, single nucleotide polymorphism. So groups like the PGC have done a fantastic job over the last 20 years of mapping locations within the genome where risk for disease occurs, neuropsychiatric disease, neurodegenerative, etc. In psychiatry, some psychiatric disorders like PTSD, depression, most of this risk tends to reside in non-coding areas of the genome. So the role that those SNPs may be playing is speculated to involve regulation of gene expression, but being in non-coding parts of the genome and being in some cases very distal to particular genes makes it difficult for us to understand how those particular SNPs may be affecting change, may be affecting disease state when you develop PTSD. And so my lab has taken the approach of trying to understand how changes in chromatin accessibility may facilitate, at least in a disease state, how SNPs may be regulating gene expression and thus causing psychopathology. So a couple of different things that can occur within the genome in terms of chromatin changes. There are local histone modifications. So the histones are the proteins that DNA wraps around. There can be changes in the different post-translational modifications. These can have an effect on whether chromatin is open or closed. Many of these SNPs are located in areas that we know should be regulating gene expression, things like enhancers, promoters, or insulators, things like that. And these may have their own unique chromatin environment. So there could be changes in histone modifications, there could be changes in accessibility, there could be changes in the underlying DNA methylation. And finally, an area that's I'm going to talk about quite a bit is how the genome itself loops and how physical genome architecture actually changes within cells to regulate gene transcription. We believe that 3D chromatin looping is a way in which distal SNPs may be brought into closer proximity to transcription start sites or promoters or insulators or what have you in terms of regulatory regions to affect gene expression changes. And so for those of you who are unfamiliar, first I'm going to talk a little bit about chromatin accessibility. So this has become, I think, a gold standard in terms of understanding chromatin regulation at a single cell level. This is mostly due to the fact that there are commercially available kits that allow you to probe both gene expression and chromatin accessibility. Quite, really, really basically, chromatin accessibility is measured in a sequencing manner. The hyperactive TN5 transposase is inserted into the genome, and you're able to PCR areas of open active chromatin that you can then sequence and map back. So what we are able to determine from those sequences that we get back are putative cis-regulatory elements. These are regions of the non-coding DNA that are predicted to regulate gene transcription. And so I've outlined a few of them here, including promoters, enhancers, insulators, which we think do the opposite. These would have, of course, shut down transcription, active enhancers, promoters that are maybe inactive. But all of these can potentially be open or closed if you're looking in a disease state and can be picked up by looking at the ATAC-seq data. In this case, we've done single-cell ATAC-seq sequencing. We can also annotate these cis-regulatory elements and we can characterize them. So we have bulk, thanks to efforts from groups like the PsychENCODE project, which have done a fantastic job of mapping CRE annotations within the brain. We're able to see whether these are novel cis-regulatory elements. Are they disease-related? Are they new? They just haven't been picked up before. And we can start to make predictions based on their accessibility and based on nearby gene expression about whether gene expression is changing due to a change in that particular chromatin accessibility. And so in our paper last year, we showed this in the human PTSD dorsal prefrontal cortex. First of all, these are the cis-regulatory elements that we identified. We identified about 500,000 cis-regulatory elements, and we were able to link the overwhelming majority of the differentially expressed genes that we detected in the dorsolateral, in the DLPFC to a putative cis-regulatory element, so that we are capturing how gene expression is being controlled from the chromatin accessibility nearby. And I'm highlighting that here in panel G for probably the hallmark gene of PTSD, which is FKBP5, where you can see that there's extensive chromatin klooping, and you can see changes across cell types for open and accessible chromatin accessibilities, which are likely driving expression of FKBP5 across a variety, in this case, across a variety of non-neuronal cell types. Probably most predominantly, we found that we were seeing a high degree of regulation in endothelial cells of people with PTSD. We can also look at whether there are disease-specific changes in chromatin accessibility, not just the gene expression itself. So when we calculate differentially accessible regions, I'll call those DARs for the remainder of the presentation, we are able to find that there are both neuronal and non-neuronally specific PTSD DARs. For the most part, we find that most DARs are lost chromatin accessibility. So the overwhelming majority of them are lost in PTSD and not gained. Lost chromatin accessibility would be more closed chromatin, and for the most part is associated with decreases in transcription. My group and Nikos's group, who's also on the call, has shown that generally speaking, PTSD results in a decrease in the transcriptome. A lot of transcripts, especially neuronal transcripts, are downregulated. This also tends to be the case across most psychiatric disorders. When we compared the nu-N positive and nu-N negative that are lost and or gained, for the most part, they're extremely specific. So what we're seeing in the non-neuronal cell types and the neuronal cell types are very different. So there are very, very different genes that are being affected in neurons versus non-neuronal cell types in terms of what's gained and what's lost and how epigenomic shifting is occurring in those particular cell types. So in our previous work, we were able to use differentially accessible chromatin to find map GWAS SNPs for PTSD. This is from an older GWAS. This is the MVP GWAS from a few years ago. And we were able to find map all of the SNPs that they identified in that particular study, most of which we were able to confirm as the likely causal SNP. In a few cases, we nominated what we thought might have been a better SNP to look at because we felt it, we gave it preference because it fell within a specific chromatin accessibility change. So we identified an accessibility change in PTSD. We placed the SNP. The SNP was placed there. We called it as we think is the causal SNP. I'm going to return to this point in a couple of slides, but I do want to point out that some of them we verified and some of them that we nominated new SNPs. So what might be going on within these more accessible or less accessible regions? Well, just because you're more open or more closed doesn't mean that you're necessarily going to increase your closeness, your proximity to other genes that the SNPs within your open chromatin region might be able to regulate. So it's become more apparent over the last 15 years that the genome is actually very three-dimensional in structure, that there are actually chromosomal territories where some parts of chromosomes are much closer to the periphery. They tend to, of the nucleus, they tend to be less active. Some regions are closer to the center, they're more active, and they fall into different compartments, the A and B compartments. B, closer to the periphery, are turned off. A are more likely to be active. And we know that there are areas where there's extensive chromatin looping, chromatin-chromatin contacts that form these TADs, these topologically associated domains. And these are basic regulatory units of the genome where extensive chromatin-chromatin interactions are occurring. And there's a more or less stereotypical structure that occurs at these particular loops. And we know that there are particular proteins like cohesin and CTCF, which is a transcription factor, which are generally bound at these particular loops. And so what we decided to do was to take our dorsal PFC data, the same samples that we looked at in our previous paper, to see if we could detect changes in chromatin looping in these same cell types. And we did do that. And what we found was actually somewhat surprising to us. We found that the overwhelming number of changes in chromatin contacts and looping were occurring almost exclusively in neurons. So the overwhelming majority are strengthening of looping within neurons. Very, very few changes in chromatin looping were observed in non-neuronal cell types. And we found that there was a statistically significant enrichment for these chromatin loops occurring within PTSD-specific DARs, or differentially accessible regions, suggesting that not only are these loops important, are they PTSD-specific, but they're relevant for regulating gene expression of particular PTSD genes. We previously have shown that the overwhelming majority of risk for PTSD occurs in excitatory and inhibitory neurons as a simple LDSC using the previous PGC and MVP GWASs. We also find that these GWASs predominantly enrich in PTSD-specific DARs. They are not enriched within non-neuronal DARs. They're only enriched within PTSD DARs significantly. They also fall within a variety of genomic clusters, but for the most part, they tend to be promoter or distal, which is consistent with a lot of the other data that's been shown for these types of SNPs. And so it's not surprising that we're able to find this. So I'm going to dive in and look at a couple of case specific genes that I think are particularly interesting to highlight how we're using this data moving forward. So cancel-1 is one of the most well-studied, understood genes. It's involved in neurodevelopment, it's involved in psychiatric disorders, and it's been implicated in neurodegeneration as well. It's on the 17q chromosome, and it codes a scaffold protein that's involved in chromatin acetylation, I believe. Cancel has been studied intensively by the PsychENCODE consortium. It has a neighboring gene, MAPT, which is also a putative tau in all tauopathies, so it's been implicated in Alzheimer's disease. We were able to identify significant decreases in chromatin accessibility that included these putative genes for cancel-1. This decrease in chromatin accessibility also lines up with decreases in gene expression for cancel-1 across all of these different neuronal cell types. MAD1L1 has been consistently identified across PTSD GWAS for the last 10 or so years. I'm highlighting MAD1L1 in the latest GWAS, the largest one from the PGC, where it's located on chromosome 7. We previously identified elfin-1, this gene by TWAS, doing one of the first human postmortem brain TWAS experiments. Elfin-1 is actually fairly close to MAD1L1 in the genome, and we wondered whether genetic regulation of MAD1L1 may be affecting elfin-1. So this was one of the first places that we decided to look for whether there would be changes in chromatin-chromatin contacts. In our initial study published last year, we actually identified a new open chromatin, decreased chromatin signature in intron 10 of MAD1L1 and nominated RS, I'm just going to call it 919. This particular SNP is likely being the causal SNP here. We predicted using publicly available high-C data that there were in fact chromatin-chromatin contacts between intron 10 of MAD1L1 and elfin-1. When we went back and looked at our own diagnostic-specific chromatin contact data, not only were we able to verify that we did see this decrease in chromatin accessibility, but we identified putative chromatin-chromatin loops that were changing in strength between intron 10 of MAD1L1 and elfin-1. So we believe that this regulation within this particular, so as opposed to cancel-1, where the SNPs are likely regulating the gene that we were found in, we believe that MAD1L1 is this particular SNP within MAD1L1 is actually regulating a nearby gene, and that's occurring via three-dimensional changing of the looping structure. This is also consistent with a decrease in elfin-1 transcription, and MAD1L1 is not quite so convincing, but this is also cell type specific decrease in the elfin-1 transcript in inhibitory neurons. We've followed this up with some work in animals using I'm not going to get into the model, but it's a traumatic stress model where we're able to find that traumatic stress in the model decreases transcription levels of elfin-1 and its putative receptor target, which is mGluR7. So elfin-1 is expressed on the postsynaptic side of GABAergic neurons. mGluR7 is located on the presynaptic pyramidal side. Both are decreased in PTSD, and we believe that this decrease results in a decrease in synaptic transmission, consistent with what's been reported in the neuroimaging data, which in which P of C hypofunction is thought to increase function within the amygdala across more hyperactivity. We're at Yale, so of course we like to look at rapid-acting antidepressants. It turns out that ketamine, the rapid-acting depressant ketamine within that same model will increase levels of elfin-1 and mGluR7, and it will restore inhibitory drive within the medial prefrontal cortex of these particular rodents. So I've been able to draw for you a line between a genetic risk gene, a human post-mortem brain functional genomic outcome of that particular gene, and then be able to show you that in a relevant model of traumatic stress, we're able to show that this has a behavioral readout and that we can and a synaptic readout change as well. And so this is my take homes. I think I'm probably running out of time, so I'll stop here, but I'm happy to ask any questions that anyone has. I do want to thank Xiao Ke Duan who's a postdoc in my lab. He's done all of the high C analysis work that I showed you today. So I will stop there, and I think we're not doing questions in between, so I think I'm just turning it over to Nikos. So I will stop share.
Yes, thank you, Matt. And we're going to have the questions at the end of the webinar. So, Nikos, the stage is yours.
Do you guys see it?
Yes.
Okay. Excellent talk, Matt. Hi, everyone. Following up on these themes, I'm gonna be showing you data on our approaches on using system biology to dissect and differentiate risk genes from disease genes. Also representing the System Biology Working Group and PsychENCODE PTSD BrainOmics project. And if you want to be involved in our monthly discussions, please shoot me an email. So the premise of system biology, in the effort of going away from the genetics to a more mechanistic understanding of the biology of disease, is trying to introduce molecular intermediate phenotypes to bridge the gap between genotype and phenotype. The molecular intermediate phenotypes would be multi-omic data, starting from transcriptomics, epigenetics, proteomics, you name it. The cool part about that is that we can do the biological experiments and study these molecular mechanisms in the brain region, the cell type we want, the developmental period we want to capture the particular biological question, and build models to study how genotype define those immediate molecular effects. And then we can move on studying the relationship of how those mechanisms and pathways relate to neurobiological intermediate phenotypes like imaging and things we can measure in blood that could be deployed in live humans. And then hopefully we would be more informed when we go to the more complex of all, which is behavior and the clinical symptoms. Historically, we have done this in PGC-PTSD, but our success was defined by two factors: the power of the GWAS that luckily improved a lot the last years, and the statistical understanding and the modeling we have done on available data at the time. So starting these approaches in 2020 with that GWAS, or the FRIS2 GWAS of PTSD, we identified only two genes, one in the cortex and one in the blood back then. They were very interesting, but it was clear to us that they were just providing a principle, but we had a lot more to learn. While both things improved, the sample size of the GWAS and the availability of models and data, like the ones that Matt described, we were able to start going from looking at the tissues as bulk, looking at risk genes at the level of particular cell types. And now, after the FRIS3 paper was prepared, then we went from the 2 genes to the 200 genes question, and we need to design a lot of follow-up biological experiments to understand what do they mean. We now know that multiple brain regions are involved, and other tissues that are not in the brain. We know that it involves a lot of cell types, neuronal and non-neuronal. The original signal we had with an enrichment of neuronal risk genes, like Matt said, now we see that we start getting also some aspects of oligodendrocytes, OPCs, other non-neuronal cell types. the multi-omicness is becoming important. And one recent thing that we have done the past year was to involve in the fine mapping approaches diverse ancestry, and also moving away from the adult brain-centric view of PTSD to also developing brain view of PTSD. With that one, I have some examples to show you. We have trained models in admixed African adult brains, but also in European ancestry brains. We realize, okay, there is a big convergence on genetically regulated expression and also EQTLs in this population, but there are also a lot of differences. And this was true both in the adult brain and in the developing brain. But what does this mean for discovery of risk genes? Here, an example on a very known group of pathways for PTSD that relate to the stress response and including the HP axis. When we have a more European-centric view of these pathways, and also because of the power of the signal in the 17q2131 locus that Matt referred to, we are able to detect, okay, there is the enrichment of these pathways, and we see this primary driven by CRH receptor 1, that is also in that locus, the receptor for CRH. But when we involve the other ancestries, we observe a distributed signal on more members of the of these pathways that are in different locations of the genome, and give us a more complete picture of the involvement of those pathways and make it a little bit more believable. The involvement is more believable because, with regions like that, you don't know which gene it is. Is it CRSR1? Is it MAPT? Is it cancel1, right? So this pathway approach of fine mapping give you extra aspects in your understanding. Now moving to differentiating risk genes for disease genes. The theoretical concept of why this is important is that the genetic program that, the germline genetics tell us is how this organism after conception is programmed to express molecular profiles throughout the lifespan. But the concept of throughout the lifespan has also the environmental component, by definition. So throughout the lifespan, we accumulate exposures, including exposures to stressful life events. These interact with our genome and influence that molecular program. And then at some point in our life, diagnosis is given, and that has an effect, the pathology of having that diagnosis and the symptoms and the chronicity of the disease that is common in psychiatric disorders has an effect, and the treatment, successful and unsuccessful, have themselves. So how do you disentangle this? In our study that was already published, we try to demonstrate how we can utilize that understanding into PTSD, and we had a study of 3 brain regions, more than 300 brains divided in discovery and replication, and assayed in transcriptomics, methylomics, proteomics, and in one region we had our first batch of single nucleus RNA-seq data, integrating UQ Biobank definitions of PTSD to study the plasma proteome, and using the GWAS of that moment. The first point I want to make is, if we have this single omic approach and favorite brain region type of approach, then we can rank the omics and the tissues that we study and say, okay, the best investment came in return of discovered disease genes came from the cortex, and particularly from RNA. But then if we go a closer look to the data, we realize that there are unique signals being brought up with potentially less power from other regions and other omic layers. Then to get a understanding of, okay, is there redundancy? Can one predict the other? We studied the relationship between those regions at the different omic layers and realized that those correlations at best are moderate, they are low to moderate correlations, and in omic layers like epigenetics, in this particular case was methylation, it's a similar picture if you study attack or histone characteristics. These are tissue specific and the correlation between regions is smaller. Um, Another topic that is important in PTSD is that PTSD is a big umbrella phenotype, and there is the notion of sex differences, early life adversity, and death by suicide. So, trying to disentangle that, we realized that the biggest driver of divergence from the primary diagnosed effect was sex, especially at the protein level. Females and male specific analysis diverge from the main analysis, while the childhood trauma driven signatures and suicide are closer. And but also depends on the layer of the data. If you look at proteomic data, that divergence is greater. And another one, very important in the field of PTSD is, the elephant in the room is PTSD versus MDD. Genetic correlation studies and other genetic approaches imply a very high overlap. But when we study the biology of disease, we see many, many differences. This was true in also in papers from Matt and collaborators, but also in our studies. That correlation drops, and also depends on like what region and omic layer you study. And then, but I started with the system biology. So what the different omic layers tell us and how they interact with each other. In a very initial way, we mapped all the omic data in common pathways as a way of figuring out the correspondence of features between omic layers is difficult. Mapping them to common pathways enabled us to correlate them at the pathway level. And we realized that the correlations were low at best at that level. So there is a lot of divergent information contained in this. So we said, okay, let's study this. Can we have a data-driven way to do a multi-factor analysis to get latent topics and see if there is convergent, divergent signal according to the omic layers and regions? Here I'm giving you two examples. One is this factor 13 multi-omic, multi-region factor. You could think about it as a biological clock because it associates heavily with chronological age, and we used it as a tool to show that both in PTSD and MDD there is an age acceleration. But when it comes to where the strongest signal of disease is, this factor 14 that was an RNA across the brain regions factor showed stronger disease signatures. Um, uh, those disease signatures, they have another level of coordinations. They act on gene networks, and these gene networks are true in different omic layers too, so you can integrate all the data together. And predominantly, we see that there is immune signaling happening in neurons and non-neuronal cell types that involve the axonal guidance and extracellular matrix organization and heavily regulation by glucocorticoids. To push this a little bit further by increasing power, we have said, okay, what if we study cortex but multiple brain regions of the cortex at the same time? Do we boost power? If there is a convergence signal, the hypothesis is that we will see more discovery, and this is what we observed. But this enabled us to see a very salient signature that these RNA, primarily RNA signatures, are lying in this leptomeninges layer of the cortex and layer 1, where non-neuronal cell types lie, while the proteomic signatures tended to be more deeper where more neurons are. Um, um, Moving on to is the risk signatures different than the disease ones? we realized that in the broad picture there's limited overlap, and we are trying to understand that. We found approximately 40 genes that had that property of both risk and disease, and for approximately 40 pathways. The genes are interesting, the pathways potentially make sense, but also bring up also some open questions. For example, we have the glutamatergic synapse and the communication between organelles, but we have also some more unknown developmental pathways to map. But effectively, what we moving towards in the future is to study gene-environment interactions. Here's a particular example of another fine-mapped gene in 17q2131 that shows that carriers or non-carriers of the risk SNP respond differentially to childhood trauma in the regulation of this gene. Moving now to the single-cell data, at the power of our initial studies, we observed that the signal for PTSD seemed to be more genetically driven and more in the neurons, involving primarily this 17q2131 locus, and in MDD tended to be more involving stress-regulated genes like FKBP5 and R4A1 ApoE. The correlation at this stage is even lower than we see in the bulk studies, but there are common pathways. One of the common pathways is related to glucocorticoid signaling, but in a very impressive way, there is a differential direction that was originally hypothesized by Rachel Yehuda and others. We also observe in this postmortem data a sensitive response in PTSD and a less sensitive response in MDD. So we designed biological experiments to test this. In this particular case, in IPC-derived neurons, we matured them to a level that they were mature neurons, plus we tested that they had a functional glucocorticoreceptor receptor, and then we generated signatures of cortisol-dependent binding and nuclear transcription to show that indeed this neuronal effect of differential direction we saw was indeed happening in neurons. Then, my last slide, we use these integrative cortex approaches also in the single cell data to move beyond the detecting this strong neuronal signal that we originally detected in PTSD. And we saw that indeed when we boost our power, we start realizing that a lot of these neuronal signals that we got had another counterpart in this situation. It was the communication between neurons and non-neurons. And at this stage, we detect that that communication is lowered in terms of strength and number. And the pathways that can be explained by this lower amount of interactions involve neuroplasticity, axonal guidance, cholesterol signaling, and vascular support that made us think about going to the UK Biobank data and remind ourselves that indeed the observations we had that the RNA signatures in the brain correlated mostly with plasma proteins give us a picture that this localized upper layer RNA signal that involves the communication of neurons and non-neuronal cell types potentially is the link of how the peripheral systems interact with the brain and affect a disease process. So I want to thank the lab, involving multiple layers of the analysis and a very wide range of collaborations that we are involved in. And I will wait for the questions at the end. Thank you.
Thank you so much, Nikos. And next up is Henry Bailey.
Can you all see my screen?
Yes.
Okay, thank you. Great talks, Matt and Nikos. Yeah, so today I'll be sharing some of the work that my collaborators and I have done on linking genetic variants from PTSD and resting state networks. So just as a little bit of background, when the brain is at rest, there are regions that show synchronized activity. And there's two main things that we can gather from looking at resting state brain patterns. So there's functional connectivity, which sort of tell us how different regions of the brains are connected, interconnected, and working together as networks. But the other thing that we can look at is called amplitude. So this is sort of within a spatially discrete region, what is the strength or intensity of the fluctuations going on in that brain region over time? And so putting these two things together, rather than looking at the brain as maybe like uh, discrete regions, we can look at it as like a graph of a bunch of nodes and interconnected edges of all regions that are functionally connected or maybe not so connected, depending on their functionality and what they're doing. And so why is this interesting? A lot of prior neuroimaging studies have shown that PTSD is associated with brain alterations. Particularly, we've observed structure and functional alterations to the brain. And so in this study, what we're looking at is sort of trying to characterize whether shared genetic risk factors influence both PTSD and resting state networks. So can we identify, specific singular nucleotide polymorphisms or SNPs that are affecting PTSD and RSNs jointly? So just to, as a little bit of background for what these networks might look like in practice, one of the most well-studied biobehavioral models is the triple network of psychopathology. And this network or this model has been highly implicated in PTSD and other psychiatric disorders alike. And the way it works is there are three networks that are involved. There's the central executive, which is focused on cognition and like when a person is thinking intently about some tasks, they're thinking hard about how to do something. There's the default mode network, which would be, active when a person is daydreaming or in an idle state and they're not thinking about anything intently. And there's the salience network, which sort of moderates a push-pull relationship between the executive and default mode network. And hypotheses and studies have shown that, alterations in the salience network sort of force a person experiencing PTSD into, maybe a default mode dominant state or a central executive dominant state. And so there's large disruptions in this particular functional network for people who are experiencing PTSD. And so we're sort of trying to understand the genetic basis for some of the relationship between why that is happening. So getting into the data that we used two large-scale GWASs to understand the pleiotropy between PTSD and RSNs. We primarily worked with European ancestry individuals. So for PTSD, we used the largest available GWAS, which was the PGC PTSD Freeze 3, which was the Niever one published in 2024. And then we technically had 75 GWASs, which were published from the Zhao et al. Paper in 2022. And so these 75 GWASs looked at the node amplitude that I was talking about before. So the fluctuations in the resting state activity in a person's brain in 75 different regions. So they had different dimensionalities. There was a 25 and 100 dimensionality parcellation. And essentially, it chopped the brain into 25 and 100 spatially discrete regions. And just as a note, some of the analyses that we did in this paper required the samples to be independent. And because the Zhao et al. And the Nievergelt et al. GWASs used UK Biobank data for the analyses that required sample independence, we dropped UK Biobank out of the PTSD GWAS and used a sample that didn't include anybody from UK Biobank. So some of the methods, you can sort of think about these as a pyramid structure. We started broadly by trying to understand the shared genetic architecture across the genome using genetic correlation and followed up by a tool called Mixer. Mixer allows us to relax the assumption that genetic variants would have the same effect direction that is used by LDSC and gets at a more nuanced association between genetic overlap in two traits. So once we understood the broad genetic architecture relationship between PTSD and the resting state networks, we were able to use a tool called conjunctional FDR, which helped us to identify specific variants that would be jointly associated between both traits. And, we could use FUMA to functionally annotate those and get an understanding of what these exact variants are doing. Um, but so as conj FDR is allowing us to understand, these variants that are jointly associated between the two traits, the next question would be, can we tell if these variants are at a single causal locus or they just happen to be jointly associated between both traits? So using the variants that we identified from conjunctional FDR, we ran a co-localization analysis to understand the causality of each of these variants at a genomic locus. And then finally, the last thing that we were interested in was like a broad hypothesis, which was, can we understand whether or not there is some direction of causality between these traits? So, is there a genetic predisposition for developing PTSD based on the amplitude of particular nodes in your brain? And so we did that through two sample Mendelian randomization. So first, the first thing that we looked at was, the genetic correlation, and what we found was a pretty complicated relationship between the resting state networks and PTSD. So 8 of the 75 analyzed traits had significant genetic correlations, but half of them were negatively correlated and half of them were positively correlated. And this might make sense because I didn't mention this before, but each of the 75 traits, they belong to different functional networks, but a lot of them are shared. So they can also be a part of multiple functional networks at a time. So maybe it makes sense that we're observing, for some networks and regions, we see negative correlation between the variants with PTSD in the networks, and for others we see positive. And so one thing to point out, is that these top three associations that we see those nodes are all part of the motor network, whereas three of the positively correlated node amplitude traits that we analyzed were part of the default mode network. So this sort of gets at this like large scale pattern of a complex association between these networks and PTSD, and there's not like a simple understanding of, when someone is experiencing PTSD, we see, decrease activity in these networks, or we see broadly increased activity in these networks. It sort of is network dependent and maybe also region dependent. And so building on that complexity, when we look at the mixer results, we also see a more complicated relationship as well, because we found 8 of the amplitude traits from genetic correlation were significantly associated. But building on that, Mixer identified 24 additional nodes that were found to be associated. And this was done by relaxing the assumption that these variants needed to have the same effect direction and allowed them to vary. So that would look like, you have a variant in, for example, the amplitude GWAS, which is like associated with increased amplitude, but then you have a variant in uh, the PTSD GWAS that could be associated with decreased risk, and you have a bunch of those all together, which allows the genetic correlation to sort of not really be able to be analyzed or picked up by LDSC, but Mixer sort of relaxes it and allows that to happen. And the Mixer results can be interpreted, to some extent, but it's a little bit difficult because we don't have a consistent shared effect direction. So we just know that these nodes have some shared genetic architecture, but characterizing the effect direction of that is hard. So zooming in a little bit to find the specific genetic variants that could be jointly associated, we looked at the conjunctional FDR results, and we found 81 unique jointly associated loci across 54 of the node amplitude traits. And so what that means is like we have 54 of the 75 traits are sharing at least one jointly associated locus with PTSD. Some of them are more significant than others. And like we saw in the mixer analysis, the extent to which those effect directions agree or disagree is not exactly consistent, and it's very much network and SNP specific. And so what you'll see here is that some genes like BPTF were identified in the Nievergelt GWAS as being PTSD associated, but some of these weren't exactly characterized in that paper. And so there's some deeper thing going on with their relationship to the resting state networks. Another characteristic that I just wanted to point out here was that you'll notice that there are repeated genes that are coming up here, and because they're spanning multiple node amplitudes and multiple functional networks. And so what we see, which I'll show on the next slide, is that, for some of these variants and some of these joint associations, they're confined into certain networks, whereas others span large amounts of functional networks across the brain. So this is a pretty large plot, but what I really just wanted to hone in on here is that some of the genes that we saw on the table, like BPTF, C17, RF58, they span a lot of these functional networks, like the attention network, central executive, default mode. And so they have a broad effect across the brain. Whereas other ones that we look at, for example, like MAP3K14, are confined to the triple network of psychopathology that we were looking at before. So they're mostly associated in the salience, default mode, and central executive network. And so, this builds on this complicated association where, the effect that a variant might have could be, broadly affecting the brain connectivity and amplitude as a whole, or it could be confined into a specific brain region and functional connectivity or functional network model. So building off of the conjunctional FDR results, we wanted to understand, which of these are associated jointly at a causal locus. In doing so, we found 17 LD-independent causal loci. And again, some of these could be associated across multiple nodes, like we saw in the previous plot. So some of them are confined maybe to just one node, but a lot of them are confined to multiple and span a a large region of the brain. The two sample Mendelian randomization results didn't come out as well as we would have hoped. What we wanted to really see was that we were able to find some causality between a person's genetic risk for PTSD and their genetic risk for the node amplitude traits. We identified three nominally significant results, which all sort of were in different areas of the brain and were members of different functional networks. But we can't really be super sure about the causal interpretations here because these didn't survive a multiple testing correction across each of the traits. And our main hypothesis for that is that we're probably underpowered to detect a causal relationship if one should exist. Because even though the PGC-PTSD freeze GWAS is big and the largest one available, the polygenicity of PTSD as a disease makes it difficult to sort of characterize genetic influences that are causing another trait. So the conclusions that we have here, we identified a widespread shared genetic architecture, but it was highly complex, and it's difficult to interpret, at a broad level what's going on. But what we can interpret is, for specific node amplitudes and for specific functional networks, here's what tends to happen in terms of the relationship between PTSD and those networks. We identified 17 specific independent causal loci. And these are, interesting specific genomic targets that could influence both brain synchronization and PTSD risk. And so would be interested to look at those a bit further. And then, as I was saying, the causality, we need to investigate this a little bit more, but it's a little bit difficult given the restrictions on sample size. But in the end, what we would want to do is maybe develop some sort of polygenic risk score. But to do that, we probably need to be able to tell what is the causal direction of risk. Like, is it PTSD causes differences in these amplitude traits lower or higher, or do genetic predispositions of lower amplitude or higher amplitude affect a person's ability or likelihood of developing PTSD after a traumatic event? Just want to acknowledge the funding that was used for this project and all of my collaborators, in particular Raj, who couldn't be here today to present, and Mark Loeb, my primary research advisor. And this paper is in preparation right now, but has not been submitted yet. Thank you.
Thank you so much, Henry, and to all our speakers. So I invite everyone to put your questions to our panelists in the Q&A box. And And I think I can start with a question. So, um, and as Hannah mentioned about like polygenic risk scores, and we see like now that we started to integrate this multiple layers of genetics, neuroimaging, transcriptomics. How do you see the generation of next polygenic risk scores will be influenced by that? Like, how do you think adding all these new layers can improve the new? I don't, probably they're not going to be called polygenic now. They're going to include like many biological omics data. So
Do you have who to who are you asking?
just overall, just to promote the discussion.
I would say like my feeling is that that would be the direction, but but there is a thing like what will have more power versus what would be more meaningful. I think potentially it will take time for us to use the right biological data to have as a good performance as the genome-wide SNP polygenic risk scores. But I think eventually maybe this could be better risk scores for stratification. Maybe they tend to be more reliable across populations. So that at least my hope.
Thank you. Do we have any questions from the participants? Yes. I guess everyone just enjoyed it so much and it was so clear. Uh, mm. Okay, any other final notes that you can do you want to share with? What is your next goals and next steps in your research?
I can make a question to Matt. Maybe what is the
Mhm.
direction he thinks we're going? Is it like diving into a detailed genomic relationship like he described, or incorporating diverse phenotypes, other brain regions? What is like where should we invest more our efforts?
Yeah, I mean, I like your plan, Nikos. I think regions for sure is really important. We got to get out of the cortex because you're right. There's so much more going on in other brain regions. Other than even though I love the cortex and I spent most of my career there, there's so much more going on. Amygdala, hippocampus, even things like cerebellum and thalamus are starting to become really interesting in other psychiatric disorders. No reason to believe that they're not going to be relevant in PTSD. Um, Phenotypes for sure. One of the things that we're missing is trauma controls. People that got trauma but did not develop PTSD do not get included in a lot of our genomic studies. They are the perfect control group. We don't have enough of them. There's just not enough of those in those that we can make those relevant comparisons. I think your approach at looking at different phenotypes like childhood trauma, sexual trauma, military trauma, are likely going to have different signatures. In fact, they could diverge into different brain regions being relevant. There's some data that suggests that sexual trauma or even childhood abuses may be more amygdala, it's shaped more by amygdala function, and military trauma, it may be more cortical, things like that. And the sex specificity is going to be a real problem there because there's an overwhelmingly more males in the military group that are donating, and there's overwhelmingly more females in the sexual assault and childhood trauma. And so we're going to have to, continuing to build our brain banks and our resources to encompass all of that is really the direction that's where I see our field going in terms of answering these questions about multiomics.
I agree.
I have a final quick question. It's more technical. So the brain bank and PsychENCODE, do you integrate that with EHR or it's just rely on next of kin information for now?
Depends on the short answer. It depends on the brain bank. How is it constructed?
Yeah.
There are some brain banks You see it a lot in neurodegenerative disorders. That is, by design, it's a longitudinal study that ends with death.
Oh,
And then, go backwards, you can get a lot of information. There are brain banks that try to do that after the fact because apart from the next of kin information, you also get a connection to the medical records. And now analyzing with the NLP or other approaches, this medical record could give you a deeper view on the health of the health history. Um, but yeah, it's very diverse, and then it becomes a little bit of the topic of how we compare data from different studies. Like uh, me and Matt, we have collaborated, and we have, in a large sense, seen that there is convergence between different studies we have done, but there could be also some differences driven by, different modes of collection of those brains.
Yeah, I think a big part of this is going to be comparing people who are in ante-mortem programs that transition into post-mortem programs to make sure that our phenotyping is good. But that's going to take a long time. But it's something that like Nikos is alluding to.
awesome. So I think we are at the end of our time. Thank you all for participating. Yeah.
Thank you for the invitation. Had
Thank
a lot of
you.
fun.
Yeah,
Yes,
it was
Good seeing
great.
you.
was
Good seeing
great.
you, Nikos.
Thank you.
Yeah, same here. Bye.